Autonomous specification. The cell commitment is autonomous specification. Appropriate cells critical determination factor within the egg cytoplasm. In other words, the egg cytoplasm is not homogeneous. Rather, different regions of the egg contain different morphogenetic determinants that will influence the cell development. Often, the transcription factor that regulates the gene expression in a manner that directs the cells in a particular path of development. So the gradient helps, and that leads the differentiation, that leads the polarization, and that's what leads the directional differentiation of the different kind of cell production from a single type of cell. Here it's a picture here shown the presumptive trophoblast cell being taken out and it's growing in an isolated culture medium, and the exact same result is found similar to that in the normal development of patella with other associated cells. And in both the kind, you can clearly see in both the case you can clearly see the cilia is there in the same time interval. And here in this picture, the picture is showing a ray diagram of a spread of cell from a single cell to its multiple differentiating cells, which gives rise to endoderm, notochord, brain stem, etc., etc. So this is a linear version of your PET maps, and here a different color will show the region of the location of that ectoderm, mesoderm, endoderm. For example, as you can see here, the upper one is blue in color, is the ectoderm. In this region, you can see this ectoderm is in the color. The red one is for muscles, which is the mesodermal derivatives. So this is exactly here is the red color is also muscles, and this cells here you can see the partially yellow and partially red in color, because this cell which later on differentiate into mesodermal cell derivatives like muscles, blood vessels, etc., some kind of things. And here also some pictures. Here you see the mesenchymal muscles cells. So the mesenchyme, the endoderm, this is the repeating the different cell thread. And yeah, here is this picture. The normal back cell is being taken out and inserted towards the dorsal side, and the result that is found is asteroiding. That means the cells from the back tissue is forming the same back tissue when not transplanted, where when transplanted on the ventral side, it will lead to normal belly tissue. That means the cell is autonomous but is not determined. And in case you remove some portion of the cell, the cells is one it can replenish this lost portion of the cell. So if a cell is removed from the embryo, the remaining cells can regulate and compensate for the missing part. So this is the thing. In this case, the left portion is being dead because of the hard needle that inserted during this embryonic stage, that means the second cell stage. So the half of the portion is dead in this tissue, while other half will remain active and functioning very well. Normally, it happens. So this is basically the two things, that is your determination and your specification, that leads to abnormal changes in whether the autonomously, whether it's affected by environment, whether this determinant factor regulate all those things. In this case, the removal of a fertilized envelope of 4 cell stage, and then all these 4 cells are isolated and separated, and this all leads to the complete larva of a Platypus larva, and the normal one gives rise a larger one, but the differentiated cells gives a little bit smaller in size. It's maybe the reason because of the cytoplasm content, but all the cells we found has different morphological characters. So that's is unique, and that's even in adult this kind of differences is found. So each cell can form a smaller but normal plateau larva. That's the thing. And the four larvae derived in this way are not identical, despite their ability to generate all the necessary cell types. That means the cells are complete. They give rise complete developmental organism, a developed organism. But the thing is that the ability, but their ability, they are not identical. That's the uniqueness that is in this case you can find. Not like a cloning. Here a cell means squashed with some glass plates, so it's unable to differentiate as it used to be normal in normal condition, and this will give rise to this kind of mixing of cells. Both the upper and lower portion get smashed with each other, rather dividing in a 3D structure. It's just dividing in a single plane, and that leads to this kind of structure formation. And since site specification not so important, different cytoplasm nucleus, fused nucleus, that means. Here some of the morphogenic gradient in an egg. I told you the cells of egg cytoplasm is not that non-polar, rather it's polar because of this gradient of this determinant factors which regulate its axis formation. And you can see in this picture the gradient of biochoid in the cytoplasm towards its anterior is comparatively higher in concentration as compared to that of your gradient of that nanos particle. I mean, because a Gödel protein, Gödel cross-section factor or cross-section gradient factor in the posterior side is higher as compared to that of your biochoid. So this caused differences in gradient which leads to the different kind of cellular region in that Drosophila. This is all. Thank you, man. Oh, one more thing here it's forgetting. So here a default state for a cell is red, and what exactly done here the fluorescence gene of different color being inserted inside the cell of embryo, embryonic cells. So when the cells just started dividing due to different kind of recombinage, that gene that is being inserted, here is the gene that your Cre recombinase. You can see CRE recombinase, the Cre recombinase which cleaves and causes recombination in the alternation that leads to different amount of that particular gene expression of that fluorescence gene, and that leads to different kind of color formation. This is also one of the example. If that's found usually in brain bow, the main concept is derived from the brain bow, and this leads to the different portion of the cell differentiation, and that's it. And one more thing here, the importance of this brain bow is that it enables the researchers to study the morphology of the cells and their interaction in any tissues at any stage, and allow us to chart the developmental lineages on an individual cell from the early embryos through its progeny to their final destination. So thank you very much. This is the brain bow picture.